First of all, thank you to the organizers for allowing me to present this work. Today, I'm here to introduce a new web server called CoCoCoNet, which allows users to compare co-expression networks across a diverse set of species. My name is John, and I'm a computational science developer at Cold Spring Harbor Laboratories. If you're interested in reading more, this work was recently published in Nucleic Acids Research as of May. A little bit of background. In 1998, Eisen and colleagues were among the first to note that clusters of gene expression profiles also clustered in function. This would eventually be formalized as the idea of guilt by association, which essentially says that genes with a similar property are more likely to share a similar function. Furthermore, if these properties are conserved between species, translational functions between orthologs become possible. The recent explosion of RNA sequencing and similar technology has allowed for convenient access to gene expression profiles for thousands of genes across many different experimental setups, typically recorded in a gene by sample matrix. We say that two genes are co-expressed if their expression profiles are highly similar. So for any pair of genes, we can measure the similarity using a correlation. And of course, doing this for every pair of genes, we can generate a network where nodes are genes and edges are correlations. Specifically, in our networks, we use Spearman's correlation followed by a global rank standardization of all edges. But why stop at just one network? We can construct co-expression networks from many independent studies and average them, producing an aggregate where consistently strong co-expression stays strong and effectively removes noise. Again, guilt by association suggests that genes with similar expression profiles are more likely to share a similar function. So to assess the quality of our networks, we implement a neighbor voting algorithm on gene ontology functional groups and report an average AUROC across 3 cross-validation folds. Intuitively, AUROCs in this context is a measure of how well connected a particular gene set is within the network, with 0.5 being poorly connected and 0.7 being quite good. With this in mind, we collect almost 900 RNA sequencing datasets across 14 species containing over 13,000 samples. Assessing each individual network, we get AUROCs in the 0.55 to about 0.72 range, with an average of about 0.63, whereas the aggregates do significantly better with an average AUROC of about 0.76. All of this data is available for download as well as through our CoCoCoNet web server. All you need to do is choose a species to work with and input your set of genes. As an example, we select yeast and input a set of 231 highly co-expressed genes obtained from the Eisen 1998 paper. Generating results will yield a visualization of the co-expression network as well as a histogram of edge strengths. Here we show the top 1% of global connections subset to these input genes. One of the main features of CoCoCoNet is the fast enrichment of genes present in the network and the ability to highlight genes annotated with that function. Here, genes annotated with the nucleolus are highlighted, showcasing the visible cluster of genes associated with it. The next main feature uses Orthodb to map genes from one species to another. Here, we've taken the yeast genes and mapped them to Arabidopsis, zebrafish, and fruit fly, and again, highlight genes annotated with the nucleolus. Without too much surprise, we can see that the nucleolus is highly conserved at extreme phylogenetic distances with AUROCs all greater than 0.8. In another example, we look at the 102 genes associated with autism spectrum disorder identified by Satterstrom and others earlier this year. Of these 102 genes, 58 of which are known to be involved in gene expression regulation, or GER, and 24 are known to be involved in neuronal communication, or NC. Using these 58 GER genes as the functional group, in humans, as well as the corresponding mouse orthologs, we see that they have a high average co-expression with one another, but are not at all preferentially connected, with AUROCs around 0.5, making targeted translational studies difficult. On the other hand, NC genes have a relatively low average co-expression with one another, but are highly preferentially connected, with an average AUROC about 0.88. This suggests that there's a shared conserved mechanism between human and mouse. Of course, these are only examples of how you can use CoCoCoNet in the data. A few other potential research questions that one may ask, for example, are which genes are related to my target gene, and do those relationships change across species? When has co-expression been conserved across large phylogenetic distances? Does my gene set subdivide into clusters that are maintained across species? Or is my gene of interest co-expressed with other genes of interest in a species I don't study? With all of these potential uses, we are constantly updating and improving our data and analyses. We plan to incorporate a functional conservation metric between pairs of orthologous genes, encompass more data from more species, include tissue and cell type specificity, and incorporate a novel gene function prediction by combining co-expression with orthology. For more details on using co-expression and function prediction, be sure to catch John Hover's talk later today in the function COSI. And finally, I would like to thank everyone in the Gillis Lab for their input and testing of the web server. In particular, I would like to thank my co-authors, Jesse, Sarah, Megan, and Manthan, for all of the work and input they've put into this web server. I would also like to thank the NIH for funding this work. And of course, none of this work would be possible without all of the researchers who make their data publicly available. And with that, I'll be happy to take any questions.